Today we're going to be talking about epistasis. This is the Genetics 311 Module 2 video. So what is epistasis exactly? Well, epistasis is just an event where the alleles from one loci modify or mask the alleles from another loci, and this actually contributes to multiple genotypes. So whether it's heterozygous or homozygous dominant, they all share a phenotype. And this also disrupts the Mendelian 9:3:3:1 phenotypic ratio that you'll see later on being disrupted. So how is that possible, right? What we see here is we see a gene, the presence of a functioning gene creates an enzyme, which almost acts as a checkpoint, right? This is just an example that we won't be using, but this mouse is colorless, and with this enzyme, it passes on to being a brown colored mouse with enzyme B created by gene B, or the presence of gene B rather, it can then transfer to being a black mouse. So we're going to be using this delightful Pokemon Mudkip as an example. This is a blue Mudkip and this is a purple Mudkip. We're going to be looking at duplicate recessive epistasis, by the way. So what does this even look like? Well, we see here that we've got a blue Mudkip and a blue intermediate Mudkip, right? So these are the two phenotypes. And then this is another phenotype. This purple phenotype only occurs if we have a big A and a big B allele, not like a big A and a little b. If we have a big A and a little b, we're just gonna hit this blue intermediate. If we have a big A or no big A, then we won't even get past this and we'll just stay blue. So this is a dihybrid cross. This is what you would see in a basic Mendelian dihybrid cross. But remember, in Mendel's, it would be a 9:3:3:1 ratio. And here, because of our epistasis, we have a 9:7 ratio. All of these purple boxes, you have the big A and the big B allele present in the genotype, which means that it crosses that pathway fully and is a purple Mudkip. And these blue boxes, you only have one or none of the big A, big B alleles. So this is not a dihybrid cross. It's much simpler. It's a big A, little B, little B, little A, big B, little B cross. And because it's not dihybrid and we don't have to cross all of the alleles, we can just do some of them, and we see that it ends up being a 1:3 ratio. So that is duplicate recessive epistasis. And here are my citations. Thank you very much.